right, here we go. So this emerging tech workshop is a little bit on the trinity of like aging and longevity research, blockchain technology, and crypto, and artificial intelligence and automation. And those are probably one of the few biggest buzzwords out there, and some of them literally scream scam at you. So especially like I think crypto and anti-aging is like those two big verticals where like people think about, okay, it's like all fraud. And the speakers here on this workshop, I think their goal is to paint your picture that you see those technologies like blockchain technology, crypto, automation, they're here to stay. And so depending on your vantage point, you either think it's like a trinity of scam or it's like the trinity that will shape the future. So we have in a way like three themes in this workshop. One is like more about like big data. I mean, big data has like revolutionized like a lot of like the bits and bytes, like the Facebooks and Amazons of the world, right? And I think biomedical research has really started to embrace big data. And by big data, I don't mean like your clinical study with 100 individuals, which is like big in clinical terms, but I think we have to get a point where we have like per experiment, thousands of animals or billions of cells being tracked simultaneously. So that's like more than like one PhD student, one postdoc, like an entire team can do. So we really need like those automated tools and artificial intelligence to help you on that. The second theme of this workshop is like, in a way, community-based funding of like novel research and especially like moonshot research, like all this research that currently is not getting funded by the NIH or like all those other centralized entities that give like money to scientists. And I think blockchain mechanisms have like in the last few years made like some progress in this way of like helping to accelerate this funding and make it faster, make it more fair, so nobody has like to spend like time writing 10-page grants and have to wait half a year. So just make it faster that we can do more science and faster. And the third part is like more about like, how can we think about biomedical businesses? Are there like novel models that we can explore that are like fairer in the entire science ecosystem? And that includes like creating novel therapeutics, but also like, can we like reimagine publishing and like other verticals of the science industry that are important to us and improve them? And with that, we have like a schedule of like 2 sessions, one on AI, computation, automation, biomedical research, where we have like speakers like Michael Petr and Alex Chamaronkov, and then like on a panel discussion, we have Andreas joining us. And then we have like a second session on decentralized science and blockchain and aging research with Tim Patterson, Sebastian, and Gary, and also like a panel discussion afterwards facilitated by Stephanie from Lifespan. And once this is finished, it's a workshop, right? We want to do like a little bit with our hands. Up the stairs, there will be like a small booth by Michael where you can get like your gait analyzed and see like how are you working? Does it tell you something about your age? And on those 2 screens, we will probably have like a demo on crypto blockchain-enabled new review systems. And then also like for people interested on how actually do I get into this crypto Web3 space, giving them an introduction, like a light way of like what kind of tools you actually need. And without more to say, I will hand over the microphone to Michael Petr. Michael is actually like used to do his PhD in Morten's lab in the University of Copenhagen. He's doing his postdoc and is actually spearheading like a spin-out company called Track Bio. And he will discuss applications of computer vision and machine learning for like phenotyping model organisms, but also like doing that at a large scale. So please welcome Michael to the stage.
Thanks very much, Max. It's a pleasure to start off the Emerging Tech workshop and the conference with our company, Track Bio, that we're spinning out of the University of Copenhagen. And our mission is to automate and standardize phenotyping at scale. And our platform spans from preclinical testing all the way to clinical testing. And the tools we make use of to make the tools in our company are rapid prototyping as well as computer vision software and AI. The preclinical platform we are developing is based on the problem we realized in the lab that is very time-consuming and it costs a lot and it is very low throughput to do experiments in the lab. And they can be prone to bias. And we want to address these needs and also improve animal welfare. So the solution we made is a cloud and camera-based platform for automating behavioral phenotyping and predicting features like age. And within our platform, we can also measure these features here. So this is the workflow of our platform. So we use camera feeds to track animals in the lab, such as flies and mice, as you can see here. And with these tracks, we can predict health outcomes, such as age. Here's example output of our platform. So in the case of flies, we can automatically calculate the survival and compare it to the distance traveled. And we can use these features to predict age with our age predictor. So here's an example of work from my own PhD. So we assessed about 6,000 flies in the study, and we weren't able to achieve this scale without this platform before. So we assessed many different genotypes as well as different drug groups, and we automatically calculated lifespan as well as distance traveled in parallel. Here are our current collaborations in flies and mice. So now on to our clinical platform that Max briefly mentioned, and the problem we saw with the phenotyping in clinical setting is that gait analysis setups are often expensive and require a lot of technical know-how. And we wanted to address these needs. So the solution we made is also cloud-based to automate gait analysis and predict features here. Here's an example of the workflow of the platform and also myself in the lab being tracked. So we're currently applying this platform to a clinical trial in the lab, and we are currently seeking to collaborate with other trials as well, particularly neuromotor disorders. Here's our business model. So the idea is that we sell the hardware to labs, and the subjects are recorded, and the analysis is performed in the cloud via SaaS, and the user receives the results. So in summary, we're using computer vision and rapid prototyping to develop these platforms. And in the preclinical platform, we're automating behavioral analysis and automating gait analysis in the clinical platform. And we're currently fundraising and seeking alpha testers as well. So if you want your gait analyzed, you can come to our booth today up the stairs. Thanks very much. Here's our team, and thanks for your attention.
Michael, that was like a very quick start. Do we have any questions already from the audience?
Yeah, thank you very much, Michael.
Yes.
For the clinical studies,
Yes.
when you analyze the gait, what will be your golden standard?
So golden standard, like the phenotypes that we're...
Yeah, just to compare it to the patients or the conditions.
So right now we're collecting in one trial with older people, but we want to also assess larger age span as well as neuromotor disorders like Parkinson's. So these are the sort of the gold standards we want to address first.
Nice presentation.
Thanks.
I have a question about Drosophila. So for Drosophila, like the main time-consuming part is not to, is to flip actually food, like to change food, right?
Yes.
So are you going to automate this part? And a second question is like for locomotion, like we have them activity monitors, right? How we can check, the locomotion. So how is it different from these monitors?
Yep. So the food transfer we're not automating, and also the current systems that track movement are using infrared, as you know, but we're just using cameras to basically track them with our computer vision software. That's the idea.
What's your thoughts about alcohol watches and everything, being able to monitor gait? How are you going to kind of go to market with, since these devices are kind of already in our hands?
Yeah. In our case, we want to use sort of just a camera application, so we could use a smartphone app, for example. But this could be used in parallel with Apple Watches, for example. Yeah.
Perfect. Thank you, Michael.
Thanks.
So Our next speaker is Alex Zhavoronkov. And I mean, Alex really doesn't need like an introduction in this community. He's like one of the organizers of the event. And he's like the kick-ass CEO of In Silico Magazine. But he also has like a black belt in Taekwondo. So before he kicks my ass, I rather give him over the stage. And Alex has, I think, almost since his childhood or early years been on the forefront of emerging technologies. So I guess he's like just a perfect speaker like to continue this session. And he will be talking on automated laboratories and data generation for target discovery. Alex, the stage is yours.
Truly happy to be here, and thank you everybody for coming. Today I'm going to talk about automated laboratories, and that's a trend in our industry. So if you are in AI, and if you already have developed the AI that can generate useful hypotheses for disease, generate useful targets, and you already have clinical products, you know which data is valuable, and you want to automate and make the process much faster and better. So, and since we're an aging, we also want to take an aging angle. So, of course, all the yada yada, not an investment advice, no forward-looking statements. As a matter of fact, you won't be able to invest in silico. Here is the drug discovery process. If you haven't seen this slide, you need to learn it by heart because it takes a long time to discover a drug. So, it will take you at least a decade to take a longevity therapeutic to the market, even with favorable regulatory conditions. Here are the probabilities of success. The lowest probability of success is in early stage target discovery. That's where a lot of hypotheses fail. So you generate a lot of hypotheses, you go into preclinical model, it fails. But after you get your target more or less validated, it's more or less a mechanical process, so the probability of success here is pretty high. It's just cost and time. And then you go clinical, most of the time you fail in phase 2, predominantly because your target hypothesis was wrong. So it's very worthwhile to automate target hypothesis and improve that stage. There are many, many ways to use AI to accelerate drug discovery and many, many ways to generate target hypothesis. You can just say, hey, Google, show me the targets for cholangiocarcinoma. And it will show you a bunch. The problem is that they are not going to be novel, and they're not going to be targeting your subpopulation that you're interested in. You can also look at bioinformatics. You can you can talk to key opinion leaders. That's most of the time, that's what Big Pharma does. But you can also use transcriptomic, proteomic, and genetic data analysis and phenotype. So those 3 data types are very valuable. You can never have enough of those. If you are using AI to discover targets, that's going to be an AI-discovered target. As a matter of fact, here you've got a really amazing company called BioAge. So Christian, if you're here somewhere listening later on, big hand. So she has several phase 2 readouts coming up. So she identified the target using AI, then in-licensed the compound, and is taking it through clinical trials. If you are venturous enough and you want to start from scratch, you also identify the target and design small molecules later on using AI. That's what we do. We do both. I have one in clinical trials, so I know the value of the data now quite a bit. So how do you automate this process? How can you use AI to accelerate those stages of R&D? So what we have done at In Silico, we developed a system called Pandaomics. I know that many of you are already using it. Some of you who are a little bit slower, you are not using it yet. But currently there are just a few alternatives on the market. There is Open Targets, there is IDG, and Pandaomics integrates 60 philosophies for target discovery that we accumulated by working with Big Pharma over many, many years. So currently it integrates over $3 trillion worth of grants. It's a lot of data. So we follow the money, we link money to publications, patents, clinical trials, and then omics repositories. And make the system work for you in a very flexible way to discover targets. So now let's try to solve a disease. Let's see if I can do it here. Okay. So let's try to go after CRC, colorectal cancer. So this is how our previous version of Pandaomics works. So this shows you the data in there, shows you the omics repositories, how much attention is paid to this disease, gives you some business intelligence, gives you some data sets. You can see that there are like a lot of data sets, 800 or more data sets. They're already prioritized by the data value, by the data quality. You select controls, so norms, diseases. In every data set you choose, you can prioritize patients there, you can prioritize populations. You do very basic quality control using bioinformatics on a gene level, so that's every bioinformatician would want to do that. You then group genes into networks using our IPANDA algorithm. So you group genes into networks and look at how those networks change their state. You can prioritize those networks by function. Let's say you want to also look at some of those pathways, how they really look like and why they're perturbed, why they're activated or downregulated. Here we have the visualization of that process, so you can see which proteins in the network contribute mostly to the activation or downregulation of the pathway. You can see how the edges are annotated, the genes are annotated. You can also include some of those data sets that, or data types that you think are more credible. You can group some of those data sets together for your patient subpopulation, and then you get this matrix. So you've got a list of genes, and then you get many, many scores. So those are different philosophies for target discovery based on omics, based on text, based on financial fundamentals, key opinion leaders, even trustworthiness of key opinion leaders, tissue specificity, but those are not novel. So originally, when you show something like that to a big pharma executive, you don't want to show them novel targets, right? Because they will say, I don't know anything, mumbo jumbo, get out of here. But then, I know CIMIC, I know SMAT, but PP53, of course I know all of those targets. Yeah, they are not exactly druggable, but they are validated. So now let's look only at those that are targetable by small molecules. You can prioritize them by those that are accessible by small molecules. They are still not novel, but now big pharma executives see, I know this one, I've tried this one, et cetera, et cetera. And then you say, well, now how about we increase the level of novelty by relying less on text and relying more on omics. So you get to something like this where you've got moderate level of novelty. You've got many omics scores that point you to the fact that those genes are more implicated in a disease, and then the system allows you to go back into bioinformatics and try to formulate your hypothesis a little bit better. And if you're a VC, you can later just take a bunch of those, or a venturous startup, you take a bunch of those highly ranked genes, do a little bit more kind of human work, and you can try them out if you especially have a high throughput system. So once you know that the system like this yields valuable targets, and by the way, recently what we do at Insilico, we like to go after dual purpose targets. So that will work in a traditional pharma paradigm. So something that pharma could take into a specific therapeutic area, so in a very classical way, but at the same time they would have been implicated in one of the hallmarks of aging or more than one hallmarks of aging, or in a very specific biological process that is implicated in aging. So we actually are for program 1 and program 2 at Insilico. Currently we have 31 therapeutic programs. That's a lot for small molecules. I have one already in phase 1 clinical trials, kind of midway or late stage phase 1, all the way from scratch from target discovery, and that's a dual purpose target, targeting fibrosis or xenofibrosis or fibrosenescence. So it's actually in this list of this of the system, so of this paper. So now, how do we automate this process to make sure that you have incremental valuable data feeding your platform, making your AI better, and also validating some of those hypotheses more efficiently? So we have a workflow which has starts with sample culture. You do hypothesis generation, and you perturb the tissue. Do in vitro assays and do NGS screening at the same time. So my idea for automation for target discovery, target validation is to get very high quality phenotypic data using high resolution, high content imaging. Once you get abundant data sets that are paired matched with other omics data types, you can rely more on imaging and less on omics, but originally you need to generate a lot of omics. So also you look at methylation and methylation response. transcriptomic response, and then deep phenotypic response as well, human and animal. Currently, what I realized in the industry is that there is a lot of human data, but there is very little high-quality animal data. So we will actually generate a lot of high-quality animal data in my lab. And now you already have seen what pandaomics can do, just on a very kind of high surface level. You will see a lot of different groups presenting work out of pandaomics, or that either touched pandaomics or is using pandaomics directly. But this system can actually prioritize compounds that will be screened in the deep phenotypic response, transcriptional response, and methylation response experiments by the lab. So you can take disease samples and normal samples, image them, or do omics analysis, do target prediction, identify molecules from large libraries to test. Then you test them on those cells, and you do it all over again to imaging, transcriptomics, methylation. And you've got CRISPR screens for target validation. So why use robotics to accelerate and augment your AI? Well, first of all, it gives you higher performance for AI iteration, so you can do faster processing, you can run tests 24/7. If there is a pandemic, you can do it automatically from the comfort of your quarantine. You can lower the cost of each batch of experiments, and also you get highly consistent data so that nobody in the lab screws up. Or if your robot screws up, you know why it screwed up, and you know which robot screwed up. So you can significantly increase the quality of data and quantity. But again, my philosophy is that you need to exhaust every publicly available repository before you go robot. Many people actually don't do this. So we are building for the past 2 years a high-performance, highly automated lab. We just got this building in Suzhou. We have the first and second and third floor. We have a dedicated robotics team for over a year now, led by Haibing Wang in China, previously at Novartis and WuXi Biologics. And we, of course, have a pretty substantial team of target hunters. That's why the bow and arrow. Those are ex-big pharma professionals who work at InSilico and make magic happen. And in Suzhou, we're building an automated lab. I'm going to show you a quick video, which integrates deep phenotypic screening, methylation, transcriptional response, and also CRISPR assays. So all in one, and it doesn't have any humans in. So this area is fully protected from humans. We rely very heavily on AGVs. If you haven't heard this term, China is number one in that area right now, by far. We have robots delivering food to our cubicles in the elevator. They compete for space in the elevator. So this is kind of the very rough walkthrough of our lab. It's launching this November. So it's actually took a lot of time to design and bring the equipment in. So it has multiple elements: cell culture, HTS, NGS screening, and lots and lots of those AGVs. So we have 5 islands fully automated. And in between the island, we achieve full automation using autonomous guided vehicle robotics, single arm and dual arm robots. We use a company called Ximaging, one of our very good partners. They're probably number 2 in China in AI-powered robotics for a laboratory that also designs many of those AGVs. So we rely a lot on those dual arm AGVs. To fully automate the lab, we also took about a year to design the software to run it. It's a pretty expensive project. And probably around November you'll be able to visit it. So let me just pause for a second here. So we've got the compound management island, we've got the cell culture island, next high throughput screening island, cell imaging and analysis island, and NGS sequencing island. Actually something that you would have in a normal lab anyway. It's just high performance equipment that is allowing you for full automation. So this cell culture platform is fully automated, as I mentioned. So we use pretty standard equipment. We just automate it using robotics arms, using Hamilton robotics, Hamilton liquid handlers. This is the videos from our partner Ximaging. So they do, they're already performing field tests of the machine. Individually, each island, it can do 3D, 2D culture. We can start with patient samples. We can start with iPSCs. I have a bunch of my own iPSCs in there made from my own tissue. We have a high throughput screening platform that relies very heavily on a new technology called ECHO. If you haven't seen ECHO, it allows you to perform really high throughput screens that does kind of anti-gravity experiments. So you measure the different reactions while the droplets are flying upwards using sound. So that's why it's called ECHO. We can process approximately 500 samples a day. Doesn't seem like much, but the data that we generate from each sample is dramatic because we do deep imaging. We also do transcriptomics methylation. So this is the real robot out of Shanghai Jiao Tong University, currently being piloted in the X-imaging facility. So it can do high throughput tasks, but also you can basically take a protocol from Nature Methods and teach it even how to aspirate properly. So you can actually implement some of the pretty unique NGS tasks using this robotics. We use NovaSeq 6000 Illumina. We have fully automated. It's actually not originally available for full automation. So reagents you would need to change manually, but we also figured out how to automate this step. And basically, again, there is a human part of the laboratory, but there is completely automated part of the laboratory where no humans are allowed. So coming to theaters near you in November. So there are many questions that need to be answered for data generation using AI. So one is, do you really need the data? Because very often you already know the target and you really don't need additional data to generate more hypotheses. Actually, most pharmaceutical companies, they do not go after absolutely novel targets. They already know the target. The head of the therapeutic area will already know the target. And if I ask you, all of you will know some of the favorite targets for longevity, right? So TORs, AKTs, and many, many others, or CERTs. So which data types are most important for application? If you're developing a biomarker, if you are targeting a population with a specific drug, you will need very specific data type. How much data do you need to train a model? So very often you need to have a lot, but sometimes you actually don't need to have a lot because you already have pre-trained models. Can you reuse or integrate publicly available data? That's the question that I'm battling with every day, because currently in the public domain, there is $2 trillion worth of publicly funded data. So if you are not using that, you are basically committing a crime in a way, because the government makes it available for you and gives additional incentives to use it. So our philosophy is that we are data recyclers. If we, just like with what you have in a hotel, soap, towel, every blanket, everything else. So you need to ensure that you reuse the data all the time. How much incremental data will benefit your AI system? That's a big question that you always need to ask. Also, can you buy the data or can you produce it yourself? It's very tempting to produce it yourself. Sometimes it's much, much cheaper to buy it. And there are many, many initiatives like Longenesis, like Nebula Genomics, and many, many others that actually allow you to buy consented data. And can you produce it cheaply and consistently? And is it going to be valuable? It's very easy nowadays on a PowerPoint to put a lab and raise from investors for data generation. But you need to show that the data is useful. That's the very important philosophy. So in early days, we also partnered with Nebula Genomics and the George Church's company to try to evaluate the various properties of data in time and look at how this data could be transacted once it's generated. We even came up with different philosophies and economic models. This is actually a quite highly cited paper. I think it has like 350 citations or something like that since it got published. So there are many different types of human data. Actually, very primitive data types like pictures or hair pictures of your retina, something that's super easy and very available. And that can be generated cheaply and easily, can be extremely valuable. As a matter of fact, if you are developing an aging clock, aging biomarker, your retinal scan can give you the same accuracy as the best methylation clock out there. And it's pretty interpretable, so that's pretty cool, right? And it's almost free. So many of those data types are actually much more valuable than omics. But if I were to choose my own data type and I would prioritize one for novel target discovery, I would try to go transcriptomics and methylation. So nowadays everybody is onto methylation clock, but that's not exactly very interpretable data type, right? And you cannot currently use it for target discovery because those methylation sites are not in the islands that are regulating some therapeutically important genes. So very difficult to make a credible story out of it. So, but if you connect the epigenetic data with transcriptomic data, then you have a pathway to target discovery. And that's why we decided to focus on this. Of course, if you have really high quality data that is very easy to obtain, plus you also get a lot of imaging data, and you know a lot about the patient, and you start with healthy ones, the combination value of this data makes it a dramatically valuable data set. So it's a great idea to work with OpenID donors. So I basically donated a bunch of my own tissue to a laboratory called iCommuno. It's a company that does cell therapy in China. They're probably the best in reprogramming, by the way, so sorry, Altos. And they achieve very, very great remethylation, almost ES-compatible remethylation through their protocol. So we made a bunch of iPSC lines also from Yeva's cells. Yeva Biskup is probably here as well. So she was on the operating table. Michael Levitt, Nobel laureate, and a bunch of other people went to China, donated their cells, and now we are using that for our high-throughput laboratory experiments. So that's kind of the intro into high-performance laboratory automation for AI for drug discovery. This is my contacts. The best question gets a cup.
Alex, it's like just fascinating to see like a laboratory of that, and imagine that like in the future there are like going to be thousands of those. I guess probably now there exists a handful in the world. Or
I think like there is only one, and I just fingers crossed that I would be able to complete the field tests by the end of October. Yeah,
so that's like the Tesla like gigafactory in the biomedical space.
I slept there for 2 weeks. Yeah.
Yeah, well, there you go. We've got time for 2 questions before we move on to the panel.
How can you detect any change inside the retina in order to say that this going down again down inside of the liver? Because by methylation you can decide the exact time, the age of harm.
Yeah.
How we can, what are you looking for inside when you scan the retina?
Sure. So not exactly directly related to my talk, but retinal imaging data is one of the most abundant data types in the world, because every time you go for an eye exam, they take a standardized retinal image, and then the ophthalmologist properly annotates this data. And one of the data types that you never miss, one of the features that is always abundant, is age. So age is always there. You do not necessarily have RP, you do not have AMD or some other disease, but you always have age. And you've got high-resolution image stored somewhere. So you can use deep learning systems that will use convolutional neural nets or some other machine learning technology to predict this one feature, age. And after that, you can actually, and that's what Hout AI, Anastasia, wave your hand, she is doing really, really actively, and you should talk to her. She knows everything about ophthalmology, and she also knows skin. Then you try to cut out certain regions of the eye and see which region contributes most to the accuracy of the prediction of age, and also maybe the diagnosis. So you create those feature importance diagrams, and then you have an experienced ophthalmologist to see which area and which region and why it might contribute so much to the accuracy of the prediction, and do the same thing as you would do with a transcriptomic aging clock or proteomic aging clock. But it's just in transcriptomics, those features are genes. So this histogram can take the top most important features, put them in a biological context in the pathway, and say, okay, this is a target, oh, and this is biologically relevant. So you can get it down to targets. With methylation data, you can't. With ophthalmology data, you actually can do it better than with methylation. The accuracy of those predictions is very amazing. The best company in the world who actually owns a lot of IP there is Google, because they use the Moorfields data from ophthalmology hospitals in the UK very early, so now it's a little bit of an ethical debate, but they managed to train very, very high accuracy predictors of age, but that's just a side product. They, of course, are going after diseases.
Perfect. That was a little bit of a longer question answer.
Yeah.
Let's move on to the panel. And
so you get the cup, yeah, sorry.
I have a quick, just for people asking questions, you need to ask into a microphone because otherwise the people that are watching it on the stream cannot hear what you're saying. So please wait with your questions until you have a microphone in your hand, okay?
Perfect. We move on with the panel then, and we will have Alex on stage. We will have also Michael again on stage. And then we also will have Dr. Andreas Mund, who is like an associate professor at clinical proteomics at the Faculty of Health and Medical Science at the University of Copenhagen. And his research focuses on development and application of novel interdisciplinary concepts called deep visual proteomics, which combines advanced imaging, artificial intelligence, and single-cell mass spec-based proteomics to understand mechanisms in health and disease. And his latest paper on deep visual proteomics actually was selected for the front cover of Nature Biotech. So if you already have that laying around in your lab and look at the artwork, that's from his research. So thank you, Andreas, for joining us here as well.
Okay, that would have been nice.
So we've got roughly 30 minutes before we have like a break. So what we're going to do, I'm going to have like 4 or 5 questions, but if there are like questions from the audience that you would like to pose to the panel, maybe start a line there, and then depending how long the line gets, I will adapt to it. So maybe let's get directly into it. I mean, Alex, you already mentioned currently, like, you talked a lot about like AI and like the data side of things. If you really want to unleash the full potential of artificial intelligence, often, especially for those deep neural networks, large quantities of data is needed, right? And a lot of biological experiments are currently like still conducted like smaller sample sizes, yielding like small quantities of data, even though the breadth of the data is going to get bigger, right? You can get transcriptomics, proteomics, methylation, like from single cells now. But how does experimental design actually have to change to really get the most of AI?
So I think that first you need to look beyond your current experimental aim. So you need to think about how to generate data that will be useful for the future. So think about yourself as an MMA fighter, not just as a grappler, as a kickboxer, or as even promoter. So it's very tempting to do an experiment that has just high quality methylation data from a specific animal, just looking at age, and that's it. It's extremely important to think multi-omics, so try to get as expensive as possible and collaborate. So the key word here is collaboration, so very rapid formation of work groups that can think into the future. And you need to think about combination value of data, time value of data, relationship value of data, and you need to look at applications, because some of the applications, like for target discovery, if you discover a great target that results in a blockbuster drug, it returns billions of dollars to the investors and to the public, and also results in a lot of quality adjusted life years. So very often methylation data, as I've mentioned, is not exactly valuable for target discovery at this point in time. So if you are doing a methylation experiment, well, make sure that you get transcriptomics and get to collaborate. So right now I'm actually trying to form a consortium for large transformer neural networks. utilizing multi-omics data for different types of clocks and also different types of applications. If you haven't seen transformers, look at those, Midjourney or DALL-E. That's just text to image, but you can also do multi-purpose. So it can do a clock, it can do target ID, and can do many other things. It's extremely expensive to train and requires a lot of data. So the only way to approach that is through a consortium. So I think that consortiums and forward-looking pathway for your data generation is the key to training AI.
Well, Andreas, you're in a way, if you zoom in, you would be the expert to create the proteomic, the spatial proteomic data if people were to get together. Can you talk a little bit about that and how that fits in and how you combine that with artificial intelligence? And is your methodology, could you use that for lipidomics and other omics approaches as well to just accelerate the data collection?
Yes, single-cell omics technologies have greatly advanced our understanding of this cellular heterogeneity in complex samples. And now, only recently, we can now also study single cells in intact tissue. And that, that leads to retain spatial information, give rise to cell atlases, and basically illustrate protein abundance, distribution, and organization and of cells in complex tissues. So traditionally, proteomics has lagged behind genomics because, in part, proteomics the proteome cannot be amplified, analogous to nucleic acids. So this leads to a requirement of ultra-high sensitivity instruments. So, and now, with recent advances, particularly on the single-cell proteomics sides, we can now apply these technologies to study these complex phenomena in tissue, in human tissue.
So if we currently go from single cell, can we then soon have single tissue, where you can analyze the entire tissue at once, or what are the next steps? How does this technology improve over the next years?
Yeah, so our angle was actually, that's why we developed deep visual proteomics, so because now we can study what we call a microcosm in a single slice, from a single tissue slice from one patient by analyzing the proteomic evolution from normal cells towards the diseased tissue within one single slice. So because we retain here the single cell, even subcellular resolution, because we harvest the power of single cell or the light microscopy approach, and then we combine this with sophisticated deep learning algorithm which identifies single cells or even subcellular structures in the tissue. And then next step would be we use automated laser microdissection to cut out these regions of interest or single cells and measure the proteomic signature of these samples using ultra-high sensitivity mass spectrometry-based proteomics. And ultimately, what we can do then is we can use, and that's what Alex was referring to, we can then combine different approaches like the imaging approach and map back the molecular signature onto the tissue in order to describe the proteomic landscape across the tissue and try to understand or try to find novel or better biomarkers. On the one hand, we will be able to better phenotype the humans and eventually can also look into the drug discovery part. Yes.
So Alex's lab in the future will have spatial proteomics in
Not
there.
right away,
Not right
we
now.
can organize a separate island for that, yeah.
Perfect, there you go. So if we think about it now from the clinical side, Michael, right? It's you're almost dealing not with single cells, but single persons, single mice. How do you see technology develop around that? Sure, you always have imaging is nice because it's cheap, but do you look at other data generators that are non-molecular?
So no, for our system, we're just using camera feed, as you know. So we really want to really push this because it's, like you said, it's low cost, high throughput, and for us, we can extract many features in this way.
But then, I mean, you can also like have the combination of your visual data together with like the molecular data, right? Yes. And I think that's where like the entire picture is coming together. I think Alex just had a slide on that, right? Yeah. Where like it's like you're building an entire cosmos of an organism. What's the price point currently for that? If you say we combine all these technologies together and say we want to have like now phenotype each human being from toe to head across all the cells, what do you think? What would the price point be to do that right now?
There are a bunch of experiments like that in the world right now, even commercially available. Human longevity is one example. So if you go to human longevity, it generates 250 gigabytes of data from you every time you visit. So it's high resolution full body MRI, even higher resolution MRI of the head, full genome. Unfortunately, they haven't gone transcriptome or proteome or any other omics that will be very, very valuable for target discovery because, again, I'm not a big fan of genomic data for target discovery unless you are looking at pediatrics and rare diseases. It's not exactly something that is great for target discovery, great complementary data type. I think that the data types that change in time are the best for target discovery, specifically proteomics, methylation, and transcriptomics. But what they do, I think that the cost of, the base cost there would be about $5,000, including some basic annotation by some of the top humans. I don't want to lower the price point right now, by the way, just like ballpark estimate. So for us, every sample, depending on how many compounds you test, would probably cost about a few $1,000, also like $2.5 for like a basic panel of drugs that we also test on the cells.
But so you see that price point going down, like DNA sequencing, right? Like 20 years ago, it was like super expensive, and now you get like the $1,000 genome. Do you see that happening to all other technologies that will measure data?
Well, at the end, you need to go imaging. So what combination value of data is and gives you is that if you have multimodal data from a single sample, which includes imaging, at the end you can train AI to recognize some of the molecular changes just from the high resolution imaging data. So that's the beauty of it, right? So I think that you need to gather as much multi-omics plus imaging over time, but at the end, the imaging data is going to help you the most.
Cool, so take more pictures of all of us, and then at ARDD 2050, we can use all this data longitudinally and see what happened. At the same time, like public data is available, right? And some data is like, you can just download from the internet, but then you have like data that is like way much harder to get because like countries own the data or like private organizations, and they might have like different data formats. Some can be used for science, some not. But that's like a treasure, right? That's like the treasure that the world has nowadays, right? How do we get all this data opened up for like scientific research that everybody can use it? Do you guys have any thoughts on that? Like,
yeah, I think for us at least, each time we're collecting more data, either in the clinical side or preclinical side, we're building the models. So we think that these models can be used for different applications and therefore make it more accessible for everyone.
It starts with data sharing.
Yeah.
And model sharing actually,
yes.
right?
Yes.
That you can just reuse it.
Yes.
How does it do like in proteomics and in the in silico world?
I mean, we in particular make our data as early as possible accessible through bioRxiv, deposit our data in bioRxiv, and also try to publish open source or open access algorithms so that people can start to reproduce and apply our algorithms. Yes. Mm-hmm.
Yeah, so it's a very valuable and important topic, actually. So over the past couple decades, the governments have scared their citizens that, healthcare data is dangerous. and that they are going to protect them by siloing their data and not being able to share that data, which is pretty dramatic because if you think about it, your credit card information or your passport information is much more dangerous and people are being damaged every day by that information being stolen. But if somebody steals your genome or transcriptome plus phenotype information, well, you might find out that your papa is not your papa, but you cannot really damage you with this data, right, unfortunately. But those silos are very difficult to break nowadays. So I operate in 9 countries or regions. We have physical R&D sites. And we have to process data locally. We cannot share it. You can only share the proceeds of these experiments on data, unfortunately. One technology that can help solve this is federated learning. When you train models on the data locally and then share models. So some governments are trying to regulate even that, which is strange. But basically it allows you to completely anonymize data and only use the clinically relevant features that the deep neural networks are trained on. So currently there are a bunch of consortiums. One is run by our kind of co-competitor. I deeply admire the company called Augment. They run Melody Consortium and they share models trained on data internationally and also on different companies that are siloing the data. So federated learning is very, very promising. Also automated labs that allow you to generate data at hospital premises and get you target information right away. So the most important is targets at the end, right? You don't need to get those huge, very heavy data. Right now, you know that Amazon ships data by trucks. So they have trucks worth of hard drives being shipped because it's cheaper than doing it over fiber. I'm not kidding. So that is why it's very, very important to have AI that can process this data locally and share the valuable insights.
Wow, I didn't know Amazon was like even shipping their own data. But interesting to see, and I think all the data amount and quantity is increasing, right? So at some point, if you want to, especially biological data is huge, right? It's like it's terabytes for human or like certain genomes. Maybe one last question before we get questions from the audience. I think it was like the venture capital giant Mark Andreessen who termed the slogan "software is eating the world," right? And we're mainly talking about software right now here, but also like some hardware. But how do you think is like the skill set gonna change, like in laboratories, especially if like in the future we might have automated laboratories in universities, hopefully, right? So that you can like just take a lot of mundane work away. So currently, like people become like jack of all trades in a way, right? But will you see like people become more specialized then again, or have more time for thinking and do less experimentation? How do you think about that dynamic of like software becoming more integral into the biomedical sciences? Andreas, you wanna?
Yeah. So I mean, like Alex already pointed out, first we have to start collaborating to get all the know-how on board, and then we have to start make the software easy to use, accessible, scalable. And then in the next step, of course, we should automate as much of the processes as possible, as beautifully illustrated by Alex. And that will, of course, then free up more time for us to think, actually, and develop the next generation of technologies. But for now, it's using the strengths of the different technologies, combine them, so multimodal, multi-omics approaches, which are highly complex, through collaboration and integration, and then start making them easy, accessible, and scalable. And then next step would be, free up more time.
Almost creating like a human species hive mind for technology and that.
Yeah. But I think that both on the individual level and on the company level, it's important to embrace the MMA practices, mixed martial arts. So, in the past, you've got boxing, you've got individual different martial arts types, and then you got UFC, right? And right now, you go as a Taekwondo practitioner or as a Judo practitioner against an MMA fighter, you'll get destroyed, right? And that's a great advance in humanity. I mean, of course, people are, putting their attention into something that is completely destructive and distractive. But it gives you some philosophies that you can integrate in your research and your business. So that's why when you hire people, it's very important to hire people who are like this. So they have many domain expertise. So they're a general practitioner, but with some domain expertise that you want to extend. So we initially, when we started, we hired people through competitions. You probably remember those massive hackathons where we ask thousands of people to outperform a recent DeepMind paper in a specific task. And then we hire top three, top four, and then they hire the rest. And you train them in biology, chemistry, you train them in basic medicine, and you train them to collaborate, and you even train them to brag a little bit. So they need to be able to explain and present the work and inspire the others. So they need to become a very well-rounded fighter. And I think in longevity, you need to have this broad spectrum view to even to generate data, because otherwise you are just going to silo it or just do a purpose-specific data generation. You're not going to get as much output as you could with a broader spectrum view.
Yeah, just to add, I think, yeah, if you make tools more hands-off for the researchers, you're going to allow more time for creativity and pursuing more hypotheses than you couldn't before. So this would allow for greater opportunity for drug discovery.
Perfect, great. I think hope out of this conference new collaborations will be born and new technology will be developed. Do we have any questions from the audience?
I think we can get a question from Slack, actually.
Exactly. We also have like the online viewers. You can post your questions in
By the way,
Slack
here
channel.
is a great example of an MMA fighter. That is your Conor McGregor of longevity.
Yeah, I don't do MMA. Just,
MD, PhD, and also can do tech.
Trying.
Program and beautiful presentations. So be Morten.
Be Alex.
So we've got a question from Nick Marcoretta on Slack. Is there potential for single-cell proteomics to interrogate protein post-translational modification and by extension proteoforms?
Yes, so that's one of our major efforts is now to extend now protein abundance towards detecting different proteoforms like PTMs, phosphorylation, ubiquitination. For now, you still need to collect larger amounts of samples. We cannot do it on a single cell level as we speak now, but these are heavily invested efforts in our group, so we definitely will head towards this goal and eventually make it accessible. Yes.
Perfect, cool.
Hi, a really interesting panel. I really support the multidisciplinary approach to aging. We have to do that. And automation is really important to make sure things are consistent. And I love what you're doing in the lab. The question is, with many disease models, cell culture models are really useful. But with aging, it's difficult, right? Because we don't really have great cell culture models for aging. We can't see a cell age. It's hard to see a cell age in a dish. So what's the solution to that? What's your thoughts on panel on that?
I mean, I can maybe directly chip in because that's actually one of the major visions and missions we had with developing deep visual proteomics, because we look at the intact tissue where we use the imaging approach to do single cell imaging, extract features from these single cells, applying sophisticated deep learning algorithm, and then cut these single cells out, analyze their proteome using this unbiased mass spectrometry-based proteomics, which gives you a hint about the molecular signature of these cells, and then map them back. And what we found with this approach is that we can see, if we look across a tissue, a human tissue, that we see what we call a microcosm in the slide. So we see this proteomic evolution of the proteomic signatures in the different areas, and that we can see that actually context matters. The same type of cells in a different location shows a completely different molecular signature.
So they're like at different ages, as kind of like, it's almost like a cross-section at different ages within the same tissue.
What we can create now is these tissue heat maps, which show us if certain signatures goes up and down during aging or during progression of disease, for example. And we have beautifully shown that with
but in terms of aging of that person, it's harder to then, you have to come back to the same source.
So I'll actually add to that a little bit. So one of the best resources in the world that has been created for this purpose is the UK Biobank. So thank you, UK Biobank, thank you, the UK government, thank you everybody who contributed, because that's the most beautiful, ultimately beautiful data set where you have a lot of healthy subjects with a lot of multi-omics data linked to, of course, phenotype and individual. You've got genomes, in many cases you've got blood tests, transcriptomes, and you can access and buy this data or work with an academic collaborator and access it. So they make it beautifully accessible. I actually also must add that in addition to cells, humans have psychology. So if you follow some of my recent papers, we even built predictors of your psychological and subjective age. And I have a really fun paper coming up which combines biological data with psychological data. So, you know, very often people don't even like to be asked, when do you expect to die, right? So that's a psychological feature, right? Many of you probably don't even, like, you're afraid. And your time horizons also affect your decision-making and behavior and what you do for your own longevity and how do you behave. So even that data has to be collected for humans, because actually the easiest way to rejuvenate today is to change your psychology. And because, you know, Laura Carstensen and Helen Fong, using their social-emotional selectivity theory, demonstrated that psychological aging is plastic. And it also relates back to your physiological age and the way you behave. So I think that even this data has to be collected and traced back to omics.
Perfect. We have like one more question from the lady on the left.
Hi, I am Hilal Kremers. I'm an epidemiologist from Mayo Clinic. And thank you very much for this inspiring panel and the pioneering work you did. I have 3 comments about 3 dimensions of data. I happen to have done some AI work over the last few years. One is quantity, and one is quality, data validity, and the other dimension is follow-up, right, in the setting of aging. So I think we need to realize that what Facebook or social media gets in an afternoon, we cannot generate that amount of data over the many years, right? There's the big N problem we have, and with AI, you need a lot of data. And not the type of data that, you know, 1 billion data points on 100 individuals is not the same thing as 100 data points in 1 billion individuals, right? That's not big data. At the end, the N is still 100, right? So I think we need to recognize some of these limitations. And when it comes to what's publicly available data out there, there's this quality issue, right? So not everything, some of the data out there is junk, and kind of trying to be able to weed out the quality of the data as compared to quality. And number 3 is, you know, cross-sectional studies do not necessarily give us what we need, because it's hard to distinguish what's the cause, what's the effect, the outcome. So it's, I would love the panelists to comment about some of these 3 dimensions, you know, the volume, quality, and the importance of follow-up. Thank you.
Anyone wants to take a crack?
Yeah, everybody, I give you 20 seconds each.
Yeah, I'll go last.
I mean, regarding the cross-sectional approach, I completely agree, and you have to really follow people or, you know, animals in the lab longitudinally to really get a high understanding of the aging process in particular. So.
Now, we, for example, take with our approach the more precision medicine approach. So we phenotype an individual and follow this individual in time and space, and thereby make a specific prediction for this individual, rather than trying to find something like a generalizing thing which harbors all the issues you just mentioned, right? Data quality, accessibility, and so on. So we come more from that angle.
Yeah, so I agree. There is many more dimensions to data than just quantity and quality. Also, data in time. So time collection value of data is very, very important. And also follow-up over time of a specific healthy individual to be able to see how healthy aging, quote unquote, turns into disease. So you need to have lots of follow-up. And that brings us back to the topic of data economics. So that is a very, very important area. I think that, everybody here, if you have kids who do not want to go into biological longevity and want to go into economics, well, life data economics is a great place to start. There is a room for everybody in longevity. So data has very similar properties to money. So it has time value, it has combination value, so think of it as a poker game. So you might have transcriptome, proteome, you might have imaging data in time, but the patient is healthy, but then the patient starts exhibiting some rare condition, ALS, boom, then you got a royal flush. It's super expensive, valuable data set that you can study to demonstrate that you can trace causality in data. So coming up with those data evaluation models can actually influence how those data banks collect those data types. But my philosophy is that there is never too much data, and very often quantity fixes the quality issue because of AI, right? You can see that nowadays you can even generate high quality data using AI, synthesized from really low quality. And if you really need to have ultra high quality, you are not doing AI because you are AI yourself. You are not being exposed to ultra high quality data all the time. You need to make inferences. As a matter of fact, we don't even know if the world looks like it looks right now. Maybe it's like complete crap and just our brain is making it beautiful. So you can always improve the quality of data with AI if you already have trained networks. So I think that quality is very often overhyped. And what is important is diversity. So even like this panel, we have all males, right? If you would just start, it's very, very bad, by the way. We won't have other panels like that. If you take the data from us, you will miss females, right? So it's important to have balanced data sets. And especially in aging, it's extremely important to have balanced data set on long tails of very young and also very, very old. Very often the data is like this, right? You have a lot of data for 20 to 50 or 20 to 60, and then a few pockets end of life. But in between there is very large vacuum. So you need to have balanced data sets.
Perfect. Thank you, Alex. I think that was like a great panel on biomedical MMA from our panelists MAA. So now we give them a hand. And we're gonna have now like a break until 10:30.